Welcome to Chapter 5. It's time to begin the work that really corresponds quite highly with what's global and important at the moment to us all. So I suspect that this could be a lot of interest to most of us. We're going to begin with what the immune system is and what it's made of. What does it do? How does it function? So during this time, it's going to be a lot of definitions. I'm going to ask you to pause the video several times so that you can complete particular tasks. So this will take a little while to unfold, longer than the video itself and the length of that. So bear with me and get all the work done as we go, and you'll stay up to date. I need you to remember that each week we're having 2 to 3 synchronous times for lessons, whether we're doing it this way or whether I'm live online with you. But realistically, in order to get through the course, remember that you'll need to be putting in a commensurate amount of time outside of that also to just ticking off all of the jobs that I'm putting in the to-do list for biology. So please make sure that you're on track and on top of all of that work, and let me know if you're having trouble. The immune system is made up of both internal and external components. The external components are comprised of things like our skin, our tears, which contain lysosomes, which release lysozymes, the wax in our ears, the hair in our nose, the mucus along our respiratory surface and our mucous membranes, things like acidity on our skin surface, as well as microflora, which benefits what our bodies will be capable of doing. The internal environment is really what our blood is doing to assist us in eliciting immune responses. The components of the blood that is important are things like the white blood cells, which are nucleated, and they take a whole range of different forms. So we will unpack what some of those white blood cells are in creation of immunity. We'll name them. We'll be able to describe a few differences between them as well. I think some of the important things are that we understand that this is all about combating the notion of disease. And disease, dis-ease, the body not at ease with itself. So it's not functioning in the normal physiological, behavioural, or structural mechanisms. The first line of defence is your non-specific defence. The targets are generalised, and there are external barriers to the pathogens themselves. The second line of defence are also non-specific. They are the internal defence mechanisms, which don't differentiate between what type of pathogen's invading, but rather that just a foreign agent has entered the body. The third line of defence is both specific and it's an internal defence mechanism. By saying that it's specific, what I mean is that it will recognise the particular pathogen, mount a defence that is specific to that or particular to that type of pathogen, and it'll also be able to develop a mechanism whereby it can retain that information for any future invasions of the same pathogen. In defence against disease, we would be able to divide it out in a nice little dichotomy like this. You can see that there are the first line of defence, which is our external barriers. They are things like your skin, your mucous membrane, nasal hairs, wax, other materials like microflora on your skin. The acidity that you produce on your skin or in your reproductive tract or your urogenital tract. And then, of course, there are the internal immunological barriers, which are generally a cellular response. So as soon as you get into the internal barriers, we divide those out to a second line of defence, which constitutes the phagocytes and the natural killer cells. Phagocytes are responsible for engulfing foreign bodies, and that's why we've got this concept of it looking like a Pac-Man again. We've already looked at phagocytosis and how that functions. So the white blood cells that are responsible for that are a group that can identify that a foreign agent has entered the body, they will go about engulfing it, and then they will use lysozymes to destroy that foreign protein, and then they'll remove those particles by exocytosis. The rest of the second line of defense is the natural killer cells, which are responsible primarily for getting rid of any cells that have mutated and have become, therefore, potentially cancerous cells. That's our second line of defense. The third line of defense are our cells which are specific to the immune system. These are our T cells and our B cells. We call them that. You might also hear them referred to as lymphocytes. So T cells and B cells are T lymphocytes and B lymphocytes. The lymphocytes are white blood cells that have a very particular role. We're going to break these down further. There are three main forms of your T cells. They are your helper cells, your suppressor cells, and your cytolytic cells. And there's the B cells, which are responsible for creating antibodies and controlling a long-term immunological memory. So a few definitions that I'd like you to pause this video now and see if you already have some pre-existing knowledge about those. All right, first up is the antigen. The antigen is a protein which appears on a cell membrane surface. It's capable of causing an immune response in individuals, but not all antigens will do this. And that's because of the second point, that some are self-antigens. Self-antigens are protein markers that don't elicit an immune response in that individual. And that's because we have particular antigens that are specific to our cells. You may have heard of self-antigen markers and non-self-antigen markers when we have dealt with the idea of doing a transplanted organ between one individual and another. And the non-self-antigens are going to be protein markers that aren't naturally present in your own body, and they will therefore cause an immune response. And in order to take the cells that have been tissue typed and the antigens are as close as possible to your own body type, you have to take immunosuppressant drugs in order to stop your immune system using your white blood cells to attack those particular foreign proteins. Lastly, there are allergens. Allergens are proteins that aren't usually going to pose an immunological threat to elicit an immune response from your body. They're generally just proteins present on the surface of materials, or they can be just partial proteins themselves. Something like peanut contains a protein which is an allergen, and when that's taken into the body and to the bloodstream of someone who will have a hypersensitive response to it, they will create an immune response to that protein, detecting it as a large threat to them. If something's infectious, it means that it's transmissible from one host to another. If something is contagious, it's an infectious pathogen that is transmitted from host to host very easily. So there's a very high rate of transfer. If a disease is communicable, while it's infectious, its rate of transfer between one host and another is relatively low. Endoparasites versus ectoparasites. Endo, as we know, means on the inside. Ecto, on the other hand, means on the outside. Infectious disease definitions continue with the notion of microscopic versus macroscopic pathogens. As you'd suggest, microscopic are too small to be seen without the aid of something like a microscope or at least a magnifying lens. A macroscopic pathogen is one which is large enough to be seen with the naked eye. So very simply, again, examples like bacteria are microscopic pathogens, whereas macroscopic ones are things like a tapeworm or a liver fluke. And some more terminology. This includes the idea of what is an endemic, an epidemic, and a pandemic. An endemic is something that is present within a particular community or space, so it's contained within a particular group. So they are geographically isolated, or usually contained within a very defined population. It doesn't have to be geographical barriers. It might be social barriers. It could be something more along the lines of a community base, in other words. So if something is epidemic, epi means to go around. So an epidermis is your skin layer. It goes around. An epicenter is a big circle that you would draw that emanates from the origin point of an earthquake, for example. So epi means to go around in a particular fashion. So here we're dealing with spreads, something that spreads out within a population. Pandemic, if something is pandemonium, for example, then that is going to go everywhere. One could think that it has no geographical barriers or boundaries. We would consider something pandemic because it's global. I'd like you to consider whether or not you have any examples of diseases that are caused by microscopic agents within your knowledge bank already. Of course, the first one that comes to mind if you're thinking of virus will probably be COVID-19. I would be very surprised if we don't see some examples of that in our VCE, of course. So other than COVID-19 for the virus, can you pause the video now, please, and create a table out of this material and list at least 2 examples of diseases that are caused by each of the listed microscopic agents. If you have to look them up on the internet, go for it, but pause until you have done that. All right, so the first ones that I'm hoping you came up with were for prions, things like BSE, which is bovine spongiform encephalitis. It leads to mad cow disease, for example. Another example of a prion is called CJD, or Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease is the human form of mad cow disease. Now, viruses that we move on to next, one example that you probably gave was COVID-19. Another one would be the common cold, influenza, or measles, mumps, or rubella. So plenty of examples there. Examples caused by bacteria would include things like tetanus and cholera, the typical things like Staphylococcus aureus, which is golden staph, found widely in hospitals. Fungal diseases include things like athlete's foot and tinea. Tinea comes in a range of forms, and you can have candidiasis, which is called thrush. Protozoal diseases, the primary one that would probably wish you to investigate would be malaria. And others would include things like Giardia. Giardia is found in water. And toxoplasmosis is another brilliant one, transmitted by the fecal matter of cats. There are a range of ways that microscopic pathogens invade hosts. Some of those ways are through vectors, which are biotic carriers of pathogens, for example, mosquitoes. Others can be airborne, and that's when droplets contain pathogens and you take them in via your respiratory system. There can be pathogens in water, and typically these are the sorts like Giardia that have the capacity to evade your digestive system and end up burrowing into the internal region of your body. Some of them just stay within the technically external part of your body, which is your alimentary canal, your gut, or your digestive tract, and they can stay in there, and their effects can still be had internally as a consequence. Food is another form of a way that a pathogen can invade a host. You take an ingested material, again, it evades the digestive system, including the acids, and it has the capacity to then take off in your body, something like a tapeworm. Contact is your last mechanism for invading hosts, and it's quite a broad one because it comprises both direct and indirect contact. Direct contact is typically where you touch another person or you exchange bodily fluid. Sounds like fun. Indirect contact is through the use of something called a fomite. Now, a fomite is a non-living factor, so an abiotic factor that acts to transmit the pathogen. So, for example, someone's discarded tissue, they've honked their COVID-19 viral particles into that. Someone else picks that tissue up, then touches their face or nose and then ingests that or inhales that, picks their nose, for example, and it gets into the mucous membranes and compromises their body. So a foamate is any non-living article that has the capacity to transmit that pathogen. Obviously, with COVID-19 at the moment, we're washing our hands or we're sterilising our hands constantly, and that lets us try to destroy those viral particles before they're capable of getting into our own body. You will have noticed, if I go back to the previous slide with you, I keep these things delineated a little bit by putting prions and viruses first. They are the non-living entities of your pathogens, whereas bacteria, fungus, and protozoa are all living. So prions and viruses don't exhibit all of the characteristics of life because they're not capable of reproduction unless they're inside of a host under very particular conditions. And then they use the host's protein synthesis machinery in order to recreate particles of themselves. So because they're not technically considered living, I try and avoid use of the terms like kill the virus. What we should be talking about is denaturing their protein, and that's through the exposure to things like alcohol, ultraviolet radiation, or even just simple soap and water to remove the pathogen rather than to kill the pathogen per se. In order to carry out a case study of a disease, there are essentially about 8 points you need to know. The disease name, when we're dealing with this, is referring to its technical or medical name is usually best. So, for example, if the disease name was rubella rather than German measles, even though they're the same thing, try and find out the technical name for it. The causative pathogen, what we're dealing with here is that the bacteria, the virus, protozoa, fungus, or whichever one I forgot to say. So which of those 5 microscopic agents would it be? The life cycle is something that is considered direct or indirect. What we mean in terms of life cycle is that if it's a direct life cycle, it is transmitted from one host of a particular species to another host of the same species. Whereas if it's an indirect life cycle, it goes from the host of one species to a different species before it can re-enter that particular original species. Think of something like malaria. So in order for that to be transmitted from one human to another human, it requires a vector to transport it. So the vector being the intermediate host in this circumstance. So the first human is then, their blood is uptaken by the mosquito then passes it on to the next human. So that's an indirect life cycle. The method of transmission is what's happening when you are, what does it take to get it from one host to another? So, for example, using the malaria again, the transmission is through the female Anopheles mosquito injecting saliva containing the protozoa, which was the causative pathogen. In that case, the causative pathogen is something called Plasmodium falciparum or Plasmodium, other species of protozoa. The effects on the host, these simply are the signs and symptoms. The host response is what does the host's body do to try and combat that particular pathogen. Sometimes the answer is nothing. Oftentimes it is things along the lines of fever, for example, to try and raise the blood temperature, and that creates a hostile environment for bacteria, for example, to reproduce themselves. Also, it might promote other favourable conditions whereby you can maximise the function of your immune system. Methods of treatment, what medically can be done for an individual to try and offset the signs and symptoms, and methods of control are the prevention things that are put in place to stop an individual from getting it in the first place. So take a moment, and what I'd like you to do is, you might as well do this on COVID-19. I would like you to just summarise very briefly by filling out this chart, if you like, for disease name, causative pathogen, life cycle transmission, effects on host, treatment and control. Come back to this video once you've done that. Look things up, write them from the top of your memory, whatever you think. Okay, see you back in a few minutes. All right, now that we've finished this particular definition section, what I'd like you to do is from page 149, have a look over the recall information, and that should bring those to your mind. You've done a little bit of work on those, hopefully have enough knowledge of those, and then go to the recap questions on page 149 for 5.1. Use the textbook to help you answer these questions, because this video alone is not enough to get you through that part, but please answer those 6 questions. Once you've done those 6 questions, unpause the video and you'll see the answers. Now that you've had a chance to look at those particular answers and mark your work, read the recall for 5.2 on page 150. And now take a moment to answer the questions for 5.2 on page 151. Mark your answers to the recap questions for 5.2. The recall for chapter 5.3 on page 152 gives you information about the fungus and the protista. So read those, and then of course you're going to do the questions for 5.3. Pause the video, then come back when you're ready to mark it.